Hello, this is Unit 5. We're going to do topics 1 and 2 today. As you can see up here in the packet, we are going from page, page 4 all the way up to 13. You are going to skip this biological application and karyotyping. We'll do that in class together. I would like to remind you of what's the big deal in this unit. The big deal here is that storage and transmission of genetic information from one generation to the next occurs through meiosis. So we're talking all about how genes get passed from one generation to the next. Let's get started. Cool. So we did some of this in class. When we're talking about heredity, we're talking about the transmission of traits from one generation to the next. The field of genetics is the study of heredity and hereditary variation. Traits are passed from parent to offspring through genes. Genes are segments of DNA that code for these basic units of heredity. You can think of it like genes are the coding section of your DNA. A lot of your DNA kind of doesn't really code for anything. It's sort of just nonsense DNA. Here, genes actually code for something: eye color, hair color, how much insulin you produce, etc., etc. Offspring acquire genes from parents by inheriting their chromosomes. So we have like sort of shotgun method of like everything kind of gets pulled over. Couple differences. We have sexual versus asexual reproduction. Asexual reproduction is a single individual. There's no fusion of gametes or anything. The offspring are the exact copies of parents. We call those clones. And mutations are the only source of variation. They can produce asexually through mitosis. So there's no crossing over. There's no combination of genes or anything like that. There is some benefits to being asexually reproducing. The first is you don't have to find a mate. If you got to make more, you just make more, right? Also, you don't have to go through meiosis at all. So there's just mitosis, single type of cell. So there are some, benefits to being asexual versus sexually reproducing organisms. In sexually reproducing organisms, there's two parents. I shouldn't say male and female. I should say sperm and egg, because parents don't necessarily have to be male, female. Offspring are unique combinations of genes from these parents, and they are genetically varied from parents and from siblings. So it doesn't matter if you share the same parents, as long as you are not identical twins. Your genetic information is varied both from your parents and from your siblings. Okay, sound good? So homologous chromosomes, this is a term we've heard before. It's a pair of chromosomes with the same size, the same length, and the same centromere position, and they carry the same genetic information. That is sort of the added kind of caveat to this that we haven't talked about previously. So you can see here you have one chromosome from your father, one from your mother, and they are homologous sort of on the same chromosome. Sorry, what's up? Okay, we're going over vocab. Yeah, we're not doing like any actual physical work. We're going over vocab. And I'm making your lecture video right now, so you're going to be on the lecture video. Are you so excited?
No.
Aw. All right, cool. So we have one from paternal and one from maternal. Here, pairs. You can see they're about the same size. They're about the same shape. When they replicate, now we have sister chromatids. So these are what we call homologous pairs of chromosomes. One homologous chromosome is inherited from mom and one's inherited from dad. And they kind of pair up like this, right? Boom, 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 boom. Great. All right. So let's talk karyotypes. Here's a karyotype. It's a way to kind of lay out this chromosomal information so we can see if there's any disorders or anything that we need to be concerned about. This patient right here is female. You can tell because there's just there's two X chromosomes. If there was an X and a Y here, that would be a male. And you can get different variations of those sex chromosomes. Again, they display chromosomes pairs ordered by length and size. You have a pair of homologous duplicated chromosomes here. This is sort of what that looks like. In karyotypes, this is actually really difficult to see. This is sort of like artist's interpretation of what a karyotype is. Normally, they just look like weird little shriveled pieces of nonsense. But this is sort of a way that we visualize it to understand what's going on. So again, pairs of homologous chromosomes. Sister chromatids are what make up each line of the X. Awesome. Okay, cells and chromosomes. So we have two different types of cells in our body. The first is a somatic cell. That's our body cells. We call those diploid, or 2N, right? We have two complete sets of each chromosome. This is in your body cells, so anything that's not a sperm or an egg. In humans, we have 46. So our 2N is 46. And then we have our gametic cells. They are haploid. They have one set of each chromosome. Humans, sperm and egg, our N is 23. But you can kind of see, if we take 23 and times it by 2, we got 46. So we're taking the gametic cells and combining them. So sperm meets egg, combine those chromosomes, and now we have 46. Cool. Kind of a review. Eukaryotes have DNA that's packaged into chromosomes, and there's two types of chromosomes. We have autosomes. So those are the chromosomes that don't determine sex of the organism. And then sex chromosomes are the X and Ys. In eggs, you have X, so X 22 plus X. And then in sperm, you can either have X or Y. This is what they talk about when they say, oh, the sperm determines the gender, right? Because the eggs are always going to have that X chromosome. If the sperm has an X, then it's going to be female. If the sperm has a Y, it's going to be male. All sexually reproducing organisms have both a diploid and a haploid number. This is very important to know. All sexually reproducing organisms have both, right? So they have different number of chromosomes in their somatic cells than in their gametic cells, because we have to have the sperm and egg come together to combine. Okay, life cycle, yay! So life cycles are the sequence of stages in the reproductive history of an organism from conception to its own reproduction. So you can see here, we're going to start down over here, sperm, which is N, so it has 22 plus a sex chromosome, and then an N over here as an unfertilized egg. They come together to fertilize. Sperm meets egg. Fertilization and meiosis alternate in sexual life cycles. So here we go. It's fertilized. Now we have a zygote. A zygote is a fertilized egg. At this point, we're 2N, right? Because the sperm is N, unfertilized egg is N. Put them together, we get 2N. Remember, N is the number of chromosomes. Then that zygote goes through mitosis to make a baby. Babies are small. We got to get bigger. There we go. Now we go through mitosis again to grow. And then meiosis occurs in our egg and sperm cells to go ahead and get fertilized once more. So fertilization is when a sperm fuses with an egg. Those are both haploids. And we form a zygote, which is a diploid. So it's combining those two sets of chromosomes together. Very cool. Here's the overall process. You may notice something that's similar. Interphase. Interphase is similar or is the same for both. So you have your chromatids here. They divide. Now we have homologous chromosomes. You'll see that they kind of exchange this information. We'll get into that in a second. This is just an overview. And then they go through meiosis again to split into these daughter nuclei. All four of them are different. So let's go over it step by step. By definition, meiosis is a process that creates haploid gamete cells in sexually reproducing diploid organisms, right? So it produces a cell that has half the amount of chromosomes in it compared to the rest of the body. What this does is it results in daughter cells with half the number of chromosomes as the parent cell. For example, humans, our diploid number is 46. Meiosis produces sperm and egg that are haploid. So we have 46 chromosomes in our somatic cells. In our gametic cells, in our sex cells, we have 23. We call that haploid. Diploid meaning 2, haploid, it's just haploid. I don't think haploid means 1, but diploid means 2. Di means 2. Okay. Meiosis itself involves 2 rounds of division, meiosis 1 and 2. But the steps are very similar. So let's go through it. There are some key differences between mitosis and meiosis. While they're very similar, here are some key differences. In mitosis, which is what we've already been over, right? Prophase, metaphase, and prometaphase, anaphase, telophase, cytokinesis. Mitosis occurs in somatic cells. Okay, so it occurs in body cells. When you cut yourself, cut your hand or something, cut your skin, those cells grow and divide. They go through the process of mitosis in order to replicate and make more of the same. You only got to go through one division, and it results in 2 diploid daughter cells. So we are starting with a diploid cell, and we are resulting in 2 other diploid cells. Daughter cells are genetically identical to their parent cells. On the other hand, meiosis is the process of forming gametes, sperm and egg. It goes through 2 different divisions, and it results in 4 haploid daughter cells. So I guarantee you on the final exam, there's going to be a question about what would result in 4 haploid daughter cells. And you would say meiosis, not mitosis. Each daughter cell at this point is genetically unique from its parent cell. Okay, let's talk some key events. There are 3 key events in meiosis that are unique. So if you can recall the key events in mitosis, with the spindle fibers coming out, connecting to the centromeres, pulling the chromosomes apart, having the nuclear envelope disappear and then reform. All of this happens in meiosis as well. There's 3 key events that are unique. In the first one, prophase 1, which by the way, the 2 divisions are numbered 1 and 2, but the major names are the same. So it's prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, anaphase 2. Okay, so in prophase 1, something happens. It's called synapsis and crossing over. We're going to get into that. In metaphase 2, metaphase 1, the chromosomes make what we call tetrads. Those are homologous pairs of chromosomes, and they line up at the metaphase plate. In anaphase 1, homologous pairs separate. So those are like the 3 main ones. Right? This idea of crossing over, this is a huge concept that you absolutely have to understand. It is the reason why we have an increase in genetic diversity. Okay, so let's get into it. Interphase. So just like in mitosis, in interphase, the cell goes through G1, which is that growth phase. We got to make sure that we have all, twice as many parts for everything. The S phase, where a new piece of DNA is synthesized from the old, so it's copied. And G2, which is just making sure everything's in place and there's no errors. So that's, again, interphase. Okay, so here we go. Prophase 1. This is where things get different. Homologous chromosomes will pair up and physically connect to each other. You can see right over here. Here's a homologous chromosome. Here's a homologous chromosome. And they kind of like hold hands. Right? We call that a tetrad, right? Because there's 4. It's like Tetris, but with chromosomes. And once the synapsis occurs, then we are able to do a recombination of genes that we call crossing over. So it occurs at a portion of the DNA we call the chiasmata. And DNA is exchanged between homologous pairs. So you can think, synapsis comes over here, homologous pairs match up, forming this tetrad. So 1, 2, 3, 4, right? And they're kind of holding hands up here. That's where that crossing over happens. So they're like swapping DNA back and forth. Every chromatid that is produced has a unique combination of DNA. So once these 2 cross over, this piece of DNA is going to be different than this piece of DNA. That's going to be different than that. That's going to be different than that. Because they exchanged these genes randomly, it's a random kind of recombination. Then we have something that's totally unique. It's totally different from its parent DNA. So this is kind of vocab heavy, synapsis, tetrad, recombination, also known as crossing over. Chiasmata, kind of vocab heavy. There you go. Boom. Tetrad. Okay. During metaphase 1, so that's prophase 1. During metaphase 1, there is an independent orientation, right? So tetrads will line up at the metaphase plate, but it really doesn't matter which goes in where, right? You can see over here, this orange one and that orange one, they're not lined up at the same, so they're not going to end up in the same cell. And in anaphase 1, pairs of homologous chromosomes separate, right? So we have this new piece of DNA that was never existed before. We have a new piece of DNA that never existed before. You can see they kind of crossed over right there. And they're going to be pulled into opposite poles. Almost exactly the same as metaphase and anaphase from mitosis. So line up at the plate, pull apart. There you go. Sister chromatids are still attached. So telophase 1 in cytokinesis. At this point, we have pretty much a unique cell over here, right? That nuclei in cytoplasm divide at this point, telophase and cytokinesis. And now we have a haploid set of chromosomes in each daughter cell. So remember, this is haploid. So we don't have, this is N, not 2N. This is N. So it's still 23 pairs. Here's where we get the diploid. So in prophase 2, what happens is we get no crossing over. I'm sorry, did I say diploid? I said haploid. So prophase 2, there's no crossing over that happens, but the same kind of steps happen. Our spindle starts to form, nucleus starts to disappear, that kind of thing. Metaphase 2, these chromosomes are going to line up at the metaphase plate. Because of crossing over in meiosis, when the chromatids are unique, you can see here this one exchanged material with this one exchanged material with this one. So this cell is unique from this one. The top one is different than the bottom. Anaphase 2, sister chromatids separate. We move to opposite poles. Again, very similar to mitosis. And then we have telophase and cytokinesis. What we end up with is 4 haploid cells. Each daughter cell is genetically unique. So these top 2 are unique from each other, but also unique from the bottom 2. So this is how we end up. We take one cell and make it genetically different from others. It's a combination of crossing over and going through this division process twice. Okay, so here's sort of the review. Early meiosis 1, we have a parent cell, 2n equals 4. End of telophase 2, we have daughter cells, n equals 2, right? In this image. Okay, take it all in. We start here, we end up here. Okay, so how does meiosis actually lead to genetic variation? This is a huge question. This question absolutely will be on your exam. How does meiosis lead to genetic variation? The first is crossing over. This process of crossing over, this recombination of genes, produces a recombinant chromosome. They exchange genetic material back and forth, so they're kind of swapping things back and forth. Another concept that's going to lead us into our next topic with Mendel is independent assortment of chromosomes. Chromosomes are randomly oriented across the metaphase plate. There's not a specific portion that they have to go to. They don't have to pair up with anyone. They are independently assorted, so they go wherever they fit. And because of this random orientation, the types of recombinant DNA that get put into one cell versus another cell are totally random. Each can orient with either the material or the paternal chromosomes closer to a given pole. Okay, that's it. So that's how people can end up looking like more like your mom or more like your dad, is that when they orient themselves, it's kind of like random which one's going to go to a given pole. And then we have random fertilization. Any sperm can fertilize any egg so long as the chromosomes match up. Any sperm can fertilize any egg as long as the chromosomes match up. Okay, so humans, any human sperm can mate with any human egg to make a totally new diversity. It's random, right? We're not pairing up. Okay, let's get it all together because we just spent 20 minutes going over all this crazy stuff. Meiosis, followed by fertilization, ensures genetic diversity in sexually reproducing organisms and provides genetic information that plays a role in natural selection. Whew. So First fertilization occurs, first meiosis occurs, I'm sorry, and then fertilization. So remember, meiosis is only happening in our sperm and egg cells. There's no meiosis going on in our skin cells or our hair cells or in eye cells, whatever. And by doing this, it ensures diversity in sexually reproducing organisms and provides genetic variation that plays a role in natural selection. So we can talk about how meiosis actually drives evolution in the sense that traits that are more adapted to their environment are going to survive. Think like camouflage. If we had an organism that survived due to camouflage, and then all of a sudden, due to sexually reproducing organisms coming together, we have a baby that doesn't have camouflage. That baby is going to get eaten, thus playing a role in natural selection. Cool. This cellular process is driven by the interaction of subcellular components and uses free energy that is required for the growth and reproduction of living systems. Whew. What this means is that the interaction of these nuclei and these chromosomes and these components that are inside the cell, they drive this reaction required for reproduction of living systems. So let's not forget to zoom in. I guess this is what this is saying. Great, that's it. I hope you enjoyed, and I will see you in class.